Hi, I'm really excited for the opportunity to share with you today our research on using genetically encoded cavitation nuclei for targeted and cell-based therapy. Cell therapy is an emerging therapeutic approach for fighting cancer that tackles this problem by engineering cells to act as secret agents. Much like James Bond, these cells can perform complicated operations within our body and try to save it from the evil cancer cells. They can be engineered to home to tumors, sense the environment, and release anti-cancer drugs locally. So why would we want to express cavitation nuclei inside these cells? The reason is that cell therapy still has some important limitation. Much like James Bond, a secret agent cell can sometimes get distracted. For example, some small portion of our therapeutic cells might end up in the kidney or the liver instead of the tumor because of some attractive biomolecular signal that they encounter. In addition, they have limited arsenal of action mechanisms. And so in this work, we propose a solution for these problems in which focused ultrasound is used to activate only the relevant cells and reduce side effects, and cavitation is used to give our cells real firepower. Our approach for combining cell therapy and cavitation is based on gas vesicles, which are gas-filled protein nanostructures that in nature are used by photosynthetic bacteria to regulate their flotation. Our lab was able to express gas vesicles in bacteria and more recently in mammalian cells. We use the air trapped inside these vesicles as a contrast mechanism. Our hypothesis in this work was that by releasing this air, we will be also able to use them as cavitation nuclei for therapy. We started by looking at the cavitation signal using passive cavitation detection. Here you can see that the passive cavitation signal increased with pressure and is much higher than all of the controls. After validating the possibility of using gas vesicles as cavitation nuclei, we tested the possible application using two approaches. The first one was targeting gas vesicles to tumor cells with the purpose of opening the membranes and enabling either cell lysis or improved drug delivery. Here you can see a bright field image of the tumor cells and the attached gas vesicles in green. We use propidium iodide as a marker of sonoporation. Before ultrasound exposure, the propidium iodide signal was negligible. However, after exposure to ultrasound, we saw an increase in the propidium iodide signal, which was a sign of sonoporation. Next, let's look at some gas vesicle expressing cells. We co-expressed a bioluminescent reporter as a drug model. We observe nearly 50% lysis of these gas vesicles bacterial cells, and also an increased bioluminescent signal in the surrounding fluid, which was a sign of controlled drug release. We saw a very selective cell lysis in gas vesicle expression in mammalian cells. However, the size of the effect was smaller because of the lower gas vesicle expression in these early versions of mammalian cells. Let's look at some in vivo results. We're able to see gas vesicles inside the tumor using amplitude modulation with negligible signal from the saline control. The passive cavitation signal of gas vesicles was much higher than all of the control, which was the first sign of gas vesicle cavitation in vivo. In another experiment, we looked at the effect of gas vesicle cavitation on the surrounding tissue. We injected gas vesicles systemically, and 2 minutes later, after they aggregated inside the tumor, we exposed them to ultrasound. We saw a significant increase in the number of hemorrhagic and necrotic foci in the presence of gas vesicles compared to the saline injections. We're currently working on several applications of gas vesicle expression in situ. For example, here we injected a secret agent cell systemically. This cell infiltrated into the tumor and produced gas vesicles in situ. Finally, I want to thank all my collaborators and the funding agencies. Thank you very much for listening, and I'll be happy to answer some questions.